Yeah, good afternoon. I'm lucky that already David gave this presentation about the phylogenetics, and also Nico gave a nice presentation about molecular clock. So in this case, I don't have to talk too much about all of these concepts. But my thesis is about estimation of the growth rate and the time of origin of somatic clones. Uh, uh, the background, the motivation of this is, can we measure the growth rate if we have genotype data? If you have mutational profiles of single cell, can we measure the growth rate? I mean, can we distinguish if the tumor are accumulating mutations at a higher frequency because of the number of generations per time? Because I was discussing this in the question for Nico's presentation, and there's an identifiability problem when you account for the two things varying. I mean, the mutation rate, which is the number of mutations per generation, and then the number of generations per unit of time. So there's a multiplication in the formula, the mathematical formula. So in the same way, in phylogenetics, you cannot distinguish between the mutation rate and the time. You need to calibrate the phylogeny in order to account for divergent times. In the same way, we have to assume as one of these things are fixed in order to infer the other one. So in this case, we were interested in the growth rate, not the mutational rate. And that's why mutational rate was a nascent parameter, if you know what it, I mean, if you have heard this term before, which is a common Bayesian term, when you want to get rid of this parameter and then to try to infer the other ones. So we assume that we have a molecular clock here and try to infer the growth rate. So this is the intuition behind the assumption that we make, and this is the motivation of my whole thesis, basically. We have published this paper on theoretical population biology. If you have more details about the model, the mathematical behind this, which is more complex that I can talk about in 15 minutes. I mean, I will focus here on more on the inference model, Bayesian inference model, and on the results on mutational data, mutation data set, and also on the real data set that we have in the lab. In this paper, in theoretical population biology, we came up with a new model. It's a different model for the coalescence. It's different one from the one that is in BEAST, for instance, which is the most highly used software in Bayesian phylogenetics. The BEAST use three different priors for the branch length in the tree, which is the exponential coalescent prior, the skyride, and the other one, which is all the three ones suffer from the same problem. The problem is that the growth rate depends on the time scale. Our model does not depend on time scale. It doesn't matter if you measure the time in generations on the physical time, like days, years. It doesn't matter, because there's an invariant property of this process, this LBD, linear Berdek coalescence, that allows you to, basically infer the growth rate as a characteristic of the process, but not how you measure the evolution of the tumor. So if you want more details, it's available, and we're more focused on this part, the two last part of the, which is the inference, and something that is very interesting is that coalescence is very well-known model for when you have all the tips sampled at the same time point. It's different from virology when you have different time points, but here you have coalescent time, and then our model, which is accounts also for the probability of this red one. This red is the sample, and the black one are the populations. The coalescent try to do inference from the sample to the populations. So in a sense, it's like a sampling theory. In a statistic, it's like you have a sampling, you have a stratified sampling, and you try to infer the population. David mentioned this, and you have a sample inside the populations, and it is not good to extrapolate the inference from the sample to the population in a naive way. You have to do it. You have to be careful with that. And our model takes this into account by having this probability of these coalescent events, the red ones, to be included in the history of the population. As I mentioned before, our growth rate is a scalar growth rate, and since it's a division of these quantities, it does not depend on the time scale. The time scale cancel out, and then it's independent of that. That's why it's an invariant of the process, and we can measure in a very efficient way this gamma, which is the growth rate, which is defined by this quantity there. The assumption of the model is that you have a molecular clock. Remember that in order to infer the growth rate, you need to fix the molecular clock. Even if you fix the molecular clock, you can infer selection. You can infer that some populations are growing faster. They are dividing faster, more generation per unit of time, and the assumptions are like this. You can see in this graph that this gamma, when you vary this gamma, the tree topology varies completely. You have more. The branch length of the terminal branches are longer compared to the tree topology. So that's informative of the number of singletons in the number of singletons means that the private mutations for the tips. So if you have the gamma is greater, you have more growth rate and you have more singletons. And and those are the two results from the paper in theoretical population biology. We came up with two different approximations of this LBD, this new model, which is M star and M2. I didn't put the formulas. The formulas are in the paper. And you can see these ratios, these points are here are ratios of the approximation divided by the LBD coalescing, which is the exact model. And you can see that the approximations are better. This is time backwards. This is backward in time. So it's the right part is the present time, and then as you approach to the MRCA, the most recent common ancestor of the sample, the approximation they lose accuracy, but then overall they are very good. This is you can see in the left scale is 0.8 to N1.1, which is very close to 1. So it's very the approximation is really good, and we found that M at 0.8, this k, which is a parameter of the approximation, which is the parameter for MK approximation, find the best accuracy when k equals to 0.8. We did a tuning over this grid, the k over 0 to 2, where and 2 to the power 5 simulations, and we found by this norm, the Euclidean norm of these vectors, that the base was k equal to 0.8. So we use this MK approximation in order to do the inference. Why we use the inference of the approximation and not the LBD? The LBD is very hard to, it's a sad model, but it's very slow to do the inference and compute the likelihood, because the likelihood depends of next to sample. Is a whole theory about coalescence, but it's best to use deterministic coalescence approximations. And here you can see the computational time comparison between the M2 and M0.8, which is the approximations, compared to 2BD, which is in the lower part. You can see that, and this is time in milliseconds, and you can see that the faster one are the MK approximation. So M2 and M0.8, but of the two, the M0.8 are the most accurate. This is the graphical model or the generative model for the one populations. And you can see there are a lot of relationship between the gamma, the time of origin, which is the scalar growth rate, the time of origin, the coalescence time, which is the tau. It's a very complicated model because of the correlation between the model, between the parameters. So in this case, there's a whole theory about this. MCMC is very slow. If you run RISC with this kind of bird depth models where you have identifiability problems for millions and billions of iterations, 200 millions is very slow, and the state space is very large because you have also the E, which is the error model for the single cells, and you have also the mutational process, which is the phi. You can assume here we are not assuming this is another property of the model. Another good thing about the model is not dependent of the mutational model that you use. You can just use CONTRA, which has no parameters. You can use CONTRA for genotypes because there are as David talked at the beginning, there are 16 genotypes, unphased maternal and paternal genotype, and the matrix is 16 by 16. It's a very big matrix. If you reduce the number of parameters, the inference could be better. And the variance you get rid of the noise and parameters, and the inference will be better if you reduce the number of parameters. And we decided to use the most simple one, the use contour 16, which is the matrix with 16 by 16, instead of using GT, which is another method used in CellFi, in the paper CellFi. And we prove, since the gamma is an invariant, that it doesn't matter what mutational model that you use. It will give you the same exact inference for the growth rate. We showed we show here. This is the one thing that already David talked about, which is the CellFi error model. For the allele dropout, it has two parameters: allele dropout, allele imbalance, and amplification error. And the same that we use for the inference and here what we use HMC. HMC, we use it because of, as I said before, MCMC is very slow when you use on models with correlated parameters. As in this case, the growth rate, the time of origin, they are all very highly correlated. And in this case, the one MCMC move is rejected many times, even though you use, in ETH they use adaptive with Kuipers, use adaptive MCMC, trying to modify the jump of the MCMC, even though that is very slow. And HMC, Hamiltonian Monte Carlo, is a proposed very high, very distant jumps in the MCMC, and it still have a high probability of being affected. So it's very efficient. It was created by Gelman and in 2014, Andrew Gelman from Columbia University, and Hoffman in this classical paper, HMC NUTS. This algorithm, which is NUT, it stands for non-U-turn sampler. I won't go into detail, but it's a very efficient sampler. We use it in STAN. This is the result for simulated data. We simulated 500 data sets with different. This is important. I saw that IFAN used two sample sizes, 40 and 100. And in this case, we simulated 400 data set with different growth rate, different time of origin, different values for the error model, for the amplification error and the allele dropout, different sample size also. These are different sample sizes. And every time, you can see in the left part, the HMC inference for gamma is very close. Those are the map, the maximum a posteriori, the median of the posterior distribution. It's very close to the true values. This is for, as I said, a simulated data set, but very diverse. In terms of growth rate, time of origin, and you can see in the right part for the time of origin, there are two here. There are two outlier points that affects the accuracy. We measure two accuracies. The depth, we count how many times the true value is included in the 95% credible interval, which is a measure of accuracy in Bayesian. Also, we measure how distant are the map, the median a posteriori, with the true value divided by the true value, which is another type of. And we use multiple regression to see what are the most important parameters that influence the accuracy of the method. And turns out there was a question before about how many cells, and the number of cells. It doesn't matter here. What matters the most is the normalized Robinson faults of the true topology, because we are using the true topology, the multi-molecular topology for CellFi, and the number of mutations, the average number of mutations per site. So this is for the result for HMC, and this is for true data set. One is the one et al. 2014. You can see the comparison between the GTD16 and the use contour 16. There are two different mutational models with different number of parameters, but use contour has no parameters. And the GT16 has a lot of parameters, has a big matrix. You can see the CellFi paper. And in both cases, the growth rate has the same distribution or similar distribution. You can see this. Those are the 95 credible interval, and the fig line are 80 credible interval. The fig one are 80 credible interval, and the fin line are 95 credible interval. So it doesn't matter the mutational model that you use. This is for comparison between the healthy cell and the cancer. You can see the cancer one are the true, and the red ones, the growth rate is very high compared to the healthy cell, which is normal because when you suffer a driver mutation, you expect that the number of division. I mean, this is important. The selection we're talking here about the number of divisions per unit of time, the number of mutations per division, increases. So this is very intuitive, and we can also, good thing about the model is it can also model something that is in phylogenomics calls what is the curve. This is based on inference for the relative population size. This relative population size divides the population size NT divided by N, present time. So it tells you how fast the effective population size varies over the evolution of the tumor. And this is our data, for our cancer data. You can see you have different clones, different mutational clones, somatic clones. You can see the different growth rate. The metastases are the one on the left, and you can see the progression of the tumor from primary tumor, from the healthy cell, primary tumor, benign tumor, and then metastasis. They have the greatest growth rate. We can also have the posterior of the T, the time of origin. I haven't talked about the time of origin, but you can, with this data, you can build like this fish plot when you can see the evolution of the tumors. You can see the evolution of the different clones across the time, because that gives you like the time of origin, but also how fast it's growing. So it gives you like a deterministic curve that you can plot in a fish plot. This is also a very good nice about the model. And this is basically the model. I didn't put the summary, but we are grading a paper right now about all these results, and I didn't put any mathematical result because this is not published yet. But the one in theoretic population biology, the hard code math is there. If you want to look at, you can do it. It's free.
Only one question.
I have a question. So when you have a birth depth model, you sort of have a forward process, right?
So,
and that defines the likelihood.
Yeah.
And to me, that seems enough to get an MCMC algorithm running. And then there shouldn't be a need to have a backwards process like the
Yeah, there was. I always put a slide about the forward, the downside. The disadvantage of using a forward model, which is what you suggest, instead of the backward using coalescent. When you use forward, you need to use sampling. You need to account also for the sampling. You have to include another parameter, which is the sampling, with the probability of sampling 1 tips in your sample, right, in your data set. When you use coalescent, you don't need to use inclusive parameter, the probability of sampling, and this reduces the number of parameters. And when you use forward, there's another disadvantage. When you simulate, it's hard to get the right combination of parameters, the delta and the mu, the growth rate, the birth rate and the death rate, in order to see a particular number of sample tips in present time, unless you include this probability, which is also increase the number of parameter, and it's a noising parameter. Will be, you don't need it in the case of if you are interested in the growth rate.
Depends on sample size. And depends on sample.
And depends on sample size. This is also in the theoretical population biology. The sample size, the higher the sample size. Carsten Buhr has this idea, and it's a brilliant idea, actually. The sample size is correlated with the time of the MRCA in the coalescent. The higher the sample size, it draws the time of the MRCA backwards, because you need more coalescent times, coalescent internal time points in order to get to the MRCA, to the recent common ancestor. So if if you have the sample size that is correlated, positive correlated with the time of the MRCA, the time of the MRCA is biased if you don't account for these correlations in your model. This is a theoretical thing that is more deep, but
Okay, good. Thank you.